Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled Neoantigens for Personalized Cancer Vaccines, Comprehensive Identification and Effective Selection. My name is Vicky, and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes, with time for a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. And at this point, I would like to thank Personalis, who developed the content for this presentation. Personalis, Inc. is a provider of advanced genomic sequencing and analytics solutions to support the development of personal cancer vaccines and other next-generation cancer immunotherapies. Their patented accuracy and content-enhanced technology forms the foundation of all Personalis products. ACE technology improves every individual step in the next-generation sequencing process, from nucleic acid extraction to sequencing assays to data analytics. This makes it possible for them to achieve augmented coverage of difficult-to-sequence genomic regions that are missed with the use of conventional sequencing techniques. Using this comprehensive approach, they provide genomic data of the highest quality and accuracy to help biopharma drive their immuno-oncology, clinical, and biomarker discovery programs, enabling the rational design and development of effective cancer immunotherapies. And now I would like to introduce our speakers for today's event, Dr. Sean Michael Boyle. Sean is Director of Bioinformatics Application at Personalis, with broad experience across analysis and interpretation of cancer genomics data. At Personalis, Sean leads a team focused on development in neoantigen prediction, cell-free applications, tumor microenvironment analysis, and biomarker analysis for the ACE ImmunoID platform. His team is also applying these technologies through external scientific collaborations with both academic and industry partners. Prior to Personalis, Sean worked on advancing genomics and personalized genomic medicine, notably with the Dr. Mike Snyder lab at Stanford University, where he developed novel methods for analyzing and interpreting patient samples from both cancer and inherited disease. Dr. Kadar Hastak. Kadar is a field application scientist with 10 years of basic and translational experience in cancer therapeutics. At Personalis, Kadar provides scientific, technical support, and customer education on the ACE cancer product portfolio. Prior to joining Personalis, Kadar was a senior scientist at Stanford University's Division of Oncology, where he led translational research studies to evaluate the efficacy of PARP inhibitors in treating breast and other solid tumor cancer models. He also was an instructor at University of California, Berkeley. Kadar completed his postdoctoral studies at the Cleveland Clinic Foundation and Stanford University and received his PhD in the field of cancer biology from Case Western Reserve University. And now, without further ado, I would like to hand the mic over to our speakers. They may begin when ready.
Thank you, Vicky, and good afternoon and good morning to all. So for both immuno-oncology translational work as well as for neoantigen vaccine trials, it is very important to understand the complex interplay between the neoantigens, the tumor modulators, and the tumor microenvironment. Therefore, having powerful genomic and transcriptomic tools that are both accurate as well as comprehensive are needed. So today I'm going to talk about our ACE ImmunoID platform and how this platform can be used both for biomarker discovery and also aid in identifying neoantigens for personalized cancer vaccine. For today's talk, we'll primarily focus on the work which we are doing with our personalized cancer vaccine customers. So as you all know, personalized cancer vaccine development process is quite complex, right from getting good quality of DNA and RNA from tumor samples, to sequencing them, to identifying neoantigens. There are certainly challenges at the vaccine synthesis part, but at Personalis, we are focused at addressing the challenges at the front end, right from tumor samples to identification of neoantigens. So a typical cancer panel, which usually consists of anywhere between 50 to 500 known cancer driver genes, is not very effective when it comes to identifying neoantigens, or even for that matter, biomarkers. In fact, recently published papers from the TCGA group, the series of pan-atlas cancer studies, have actually shown that analysis which is focusing only on known cancer drivers and mutations for certain cancer would miss non-canonical cancer driver genes as well as mutations for that cancer and that patient. Therefore, it's very important to have broad coverage of the entire genome to identify neoantigens as well as biomarkers. So with our ACE immuno ID, we cover all 20,000 genes, and we also sequence matched normals to give you highly specific somatic variants. Along with DNA, we also use our ACE augmented technology for RNA sequencing to give you information on gene expression as well as RNA variants. Integrating DNA and RNA analysis using our powerful bioinformatic pipeline helps us comprehensively identify neoantigens as well as biomarkers. So what we consider our ACE immuno ID platform is this universal biomarker platform where we can give you information on tumor burdens. We can also give you information on neoantigens as well as HLAs. Along with that, we also give you information on antigen processing machinery, such as genes like B2M, TAP. Along with that, we also give you information on genes that are involved in DNA replication and repair pathways, such as BRCA1 and BRCA2, as well as immune modulators, such as PD-L1, ICOS, CTLA4. Now, this is getting more important because studies have shown that tumors which have defect in DNA repair and replication pathways actually have a higher neoantigen load. And recently, there has been a lot of focus which is trying to look at combination therapies between neoantigen vaccine as well as immune modulator checkpoints such as anti-PD-L1 or PDLA. Along with that, we also give you information on chemokines, genes involved in adaptive and innate responses, cytotoxicity, as well as tumor-associated antigens. So as the search continues for identifying more accurate neoantigens for developing immunotherapy, what I would like to do is walk you through a few of the key challenges that researchers are facing and how at Personalis we've tried to address these issues. So before I proceed, there is a quick poll question which Vicky has to ask the audience.
Great, thank you for that, Dr. Hasthak. And the poll question is launching on your screen right now. And that question is, what do you feel are the biggest obstacles in developing neoantigen-based therapies? And the options we have here are comprehensively identifying neoantigens, or selecting the right neoantigens, or identifying the most promising combination therapy approach, or lastly, operational hiccups such as poor samples or long TATs. So if the audience could please get their answers in, this first poll question will be closing shortly. And once again, this poll question is asking, what do you feel are the biggest obstacles in developing neoantigen-based therapies? And once again, the options are comprehensively identifying neoantigens, or selecting the right neoantigens, or identifying the most promising combination therapy approach, or lastly, operational hiccups such as poor samples and long TATs. And it does look like majority of the audience has voted, so we'll go ahead, close this first poll question, and share the results. And it looks like majority of the audience with 50% voted for operational hiccups such as poor samples and long TATs. Next was 36% with selecting the right neoantigens, and lastly 14% with comprehensively identifying neoantigens. And with that, I'll hand it back to you.
Thank you, Vicky. So it's very interesting that you brought operational hiccups as one of the key challenges. In fact, we are going to answer those in one of the 4 challenges which I just highlighted. And even Sean is also going to talk about how do we comprehensively identify neoantigens too. So let's discuss one of the initial challenges, which is the ability to work with difficult samples, such as FFPE samples when it comes for DNA and RNA sequencing. So as we all know, patient samples are very precious and limited, and traditional processes actually involve sending multiple sample preparations to different vendors to give you essential data. But we consider our ACE-immunoID as a one-stop solution where we do dual extraction of DNA and RNA in a very sample-sparing manner to give you broad data, which is important for both cancer vaccine development as well as for biomarker discovery. So in our lab, we receive a variety of different FFPE samples. This is just a snapshot, what we are showing here of what different kind of materials we get, both in terms of quality and quantity. And we actually have optimized our workflow to work with these challenging samples. And we have currently greater than 95% success rate with our FFPE samples, which we receive from our cancer vaccine customers. So how do we assess the quality of sequencing from FFPE samples? So we at Personalis look at 3 different metrics. We look at the person mapped reads, we look at the average QAM score, as well as the average QA score. And now if you look at the graph here, this is a graph of sequencing from 130 randomly selected samples from clinical trial patients. And if you see, greater than 95% of these samples had got very good high quality sequencing. So the second challenge which I would like to talk to you about is how do we comprehensively identify potential targets, both neoantigens as well as biomarkers. So it starts with improving the assay. So for biomarker discovery, broad assays such as whole exome and whole transcriptome are very essential and important. Unfortunately, the issue is that conventional assays such as conventional exomes can have very inconsistencies in coverage. So let's take an example of the gene shown here, which is the STK11 gene, which was covered using conventional exome. All the exons have been concatenated together here on the x-axis, whereas the depth of sequencing is shown here on the y-axis. Now, if you see, the depth of sequencing is around 150x, whereas the average depth is around 100x. But there are certain regions as shown here which do not have good coverage, whereas there are certain exons here which have completely missed coverage. So any variants, or for that matter, neoantigens which might be within this region would be completely missed. So just simply sequencing more or deeper actually won't fix the issue of inconsistent coverage. So we at Personalis have realized this problem and addressed this with our proprietary ACE approach. So ACE stands for accuracy and content enhanced. And the way we have tackled this problem is we still use the standard exome as our foundation. So you have broad coverage of all 20,000 genes. But then what we have gone is we've gone back and we've developed additional probes to target these problematic regions. Now, just developing additional probes is not going to solve the problem completely. So what we have also done is we've also optimized the chemistry to go along with it. And along with that, what you get is a very integrated, high accurate data set. So again, just to show you, this is the coverage which you get with conventional exome, where you have a lot of inconsistencies in coverage. And this is a nice visual of the coverage which you get with our ACE technology. Now, if you see here, these exons which were completely missed with conventional coverage now have got augmented coverage with our ACE technology. And what data you get is both blue plus green data together. And I just wanted to highlight is that we do not require any additional sample to give you this comprehensive data. All the library preparations and sequencing, everything is done in parallel for this. So as you all know, neoantigens can occur anywhere in the genome, right, including housekeeping genes. Therefore, we have actually augmented more than 8,000 medically relevant genes. So for example, let's look at the gene SYN1. And what we have done here is we've plotted all the neopeptides which can bind to their respective HLA alleles. Now, if you look at the box here, these are the potential neopeptides which bind to their HLAs, which would have been missed with conventional exome if you did not have very consistent coverage. Accurate whole transcriptome sequencing is also very critical when it comes to identifying neoantigens or biomarkers. However, with FFPE samples, the quality of RNA is not very good and it's quite often degraded. So standard enrichment protocols such as ribodepletion or poly-A are not that effective. So what we have done is we've actually used the same ACE probes which we use in our exome. And we have done an exome capture enrichment protocol to give you very uniform coverage as shown here, just like the STK11 gene. And this is the data which is generated by our approach. So if you see here, around 80 to 90% of the bases are aligned to the exonic region with our ACE transcriptome. However, if you use standard ribodepletion, only 40 to 50% of these regions are aligned to the exonic region, whereas you have a lot of off-target reads as shown here. So another critical step when it comes to having these assays is to have a very strong analytical validation. And what we have done is we have done a very robust validation in FFPE samples, in fresh frozen samples, and shown here, we've also used known cancer cell lines. And what we have done with these known cancer cell lines is we've done serial dilutions of these cancer cell lines to establish the limit of detection. And these are the results from our validation. Once again, we've done a very robust validation where we've looked at more than 16,000 SNVs and 700 indels. And if you can see, we have greater than 90% sensitivity and specificity, both for our exome as well as for our transcriptome for minor allelic fractions of greater than 10%. This is to give you a quick overview of our product configuration. We sequence the tumors at 28G, which is around 200x. We sequence the normals at 10G, which is around 70x. Our transcriptome is 50 million paired end reads, so that is a total of 100 million reads. And this entire configuration is analytically validated. We also have 3 different processing options. We offer research-only use, we offer GCLP, as well as GCLP, which is performed by clinical laboratory scientists. And this offers you a rapid turnaround time of 14 calendar days. And most of our neoantigen vaccine customers prefer having this option of GCLP performed by CLS. So another challenge which I would like to talk to you about when it comes to identifying neoantigens and biomarkers is, do we have the processes in place to enable a reliable turnaround time for data delivery? So in addition to innovating with the platform, we have also built an enterprise scale genomic management system, which we call as Symphony. And what Symphony allows us to do is allows us to track the samples right from its arrival, through sequencing, through bioinformatic analysis, and through secure delivery data. What it also allows our project manager, when you sign a statement of work with us, is to lock down the pipeline for a specific clinical trial or for a specific research project. So we know ACE ImmunoID platform is very innovative platform, But to make the platform more practical for pharma use, what we have also done is we have significantly scaled our operations. So we recently purchased the latest instrument from Illumina, the NovaSeq. We also have invested heavily in lab automation and robotics to meet around faster turnaround times. As I mentioned earlier, we have our genomic management system and also the workflows in place. We have very high performance secure computing services, as well as we have recently renewed our CLIA and CAP certification with 0 deficiencies. And what this allows us to do is this puts Personalis at a very unique position to support transformational immunotherapies as well as combination therapies that are being explored. With that, I would like to hand over the presentation to our Director of Bioinformatics and Applications, Dr. Sean Boyle.
Great, thank you very much, Kadar. So for my portion of this presentation, I'll be discussing on informing candidate neoantigen selection for more effective vaccine development. So we'll take a moment here for another poll question.
Great, thank you for that, Dr. Boyle. And this brings us to our second poll question for today. And that poll question is, beyond MHC binding affinity, what additional criteria may prove to be essential for effective neoepitope selection? And the options we have here are gene and variant expression, or similarity to self, or similarity to known antigens, or lastly, immunogenicity. So if the audience could please get their answers in, the second poll question will be wrapping up shortly. And once again, this poll question is asking, beyond MHC binding affinity, what additional criteria may prove to be essential for effective neoepitope selection? And it does look like majority of the audience has voted, so I'll go ahead, close this poll question, and share the results. And it looks like 36, or majority of the audience, voted for immunogenicity. Next was 27% with gene and variant expression. Next, it was a tie of 18% between similarity to self and similarity to known antigens. And with that, Dr. Boyle, I'll hand it back to you.
Great, thank you very much. And we really agree that looking at immunogenicity and both the gene and variant level expression is critically important. In fact, I'll be speaking on our thoughts on variant level expression here in my presentation. Great, here's an overview of our neoantigen analytics engine, where we go from our DNA cancer and transcriptome analysis modules into identification of SNVs, indels, and fusions, and really phasing those and identifying which mutations we're going to take forward into the antigen processing steps. In antigen processing, we take those events and we turn them over into potential neoepitopes. Also, within the analysis module, we assess MHC binding prediction for class 1 and class 2. We assess the similarity to known antigens, as well as similarity to self-antigens from that individual. We also assess the immunogenicity, or taking a look at the central amino acids, which are surface-facing, to see whether their characteristics are likely to be more immunogenic or not. And then, very importantly, we not only look at the gene-level expression from the transcriptome pipeline, but also the variant allele frequency in the RNA. And then all of this is delivered together in our neoantigen ID reporting. Today I'll be focusing in on 3 key approaches to neoantigen identification, including integrating the RNA, improving MHC binding prediction, and optimizing coverage of HLA alleles. So let's start off by talking about the importance of integrating RNA into the neoantigen prediction. Here again, we have an overview of our neoantigen analysis module. Again, remember, going from both the tumor exome and transcriptome, we can take FFPE samples as well as fresh frozen samples to identify those somatic events and then assess whether they're likely to be neoantigens. Now, what I want to focus on here is part of our RNA analysis module here is looking at not only the gene level expression, but also the variant level expression. This whole process allows us to go from thousands of mutations down to a handful of candidate neoantigens, which are most likely to be immunogenic. Now, here we're looking at a set of melanoma samples, which we've been working with in collaboration with Inova, a hospital system in DC. Now, what we're looking at here is the mean number of mutations which were identified in each sample. So here we see, on average, in each melanoma sample, these are late-stage melanomas, we saw 1,071 somatic variants. 709 of those were in non-synonymous. Now, if we apply a very stringent expression threshold of greater than 15 TPM, we see that that number of potential variants drops down to 268. Now, if you do a further filter, looking at RNA allele frequency of greater than 5%, we see that we have a further 30% reduction in candidate neoantigens. And this is critically important because if you stop at the expression level of the gene and you don't move on to looking for the variant evidence in the RNA, potentially 30% of your neoantigen vaccines are targeting events which aren't actually present on the cell surface. Because if it's not transcribed, it's less likely that it's going to be translated. It's also important to note that we perform an extensive validation, as Kadar had spoken on, in RNA, looking not only at small variants, but also at fusion events. The next piece I'd like to speak on would be improving MHC binding prediction. We're doing a lot of work in this area. Now, traditional approaches have really just looked to see whether a peptide can be bound and presented within the MHC pocket for class 1 and class 2. This is done through folding assays or competitive binding assays. And this doesn't really take the other pieces of the processing, which are critical to understanding whether it's going to be presented in a real cell. Now, if we look at IEDB, which is one of the widest and most comprehensive public databases available for neoantigens and MHC-bound peptides, here we see we have class 1 HLA alleles across the bottom. And then this lists the number of binders and non-binders in blue and green respectively that have been identified in each allele. Now, it's estimated that there are thousands of class 1 alleles in population. And here we see that there's binding data actually for only a handful of those alleles. And in fact, if you take a look at the number of alleles which have really a sufficient number of peptides for training, we see that that's a very small handful of alleles. Now, what we aim to do is really improve not only the number of peptides and alleles, but also to really improve the quality of the data that we're bringing into our predictions. We do this by taking in the cleaving, the transport, the loading, and the shuttling into our consideration and into the analysis we perform. And we do this by actually monitoring and assessing the number of peptides which are presented on the cell surface of live cells. In fact, to be able to define exactly which peptides are presented on each allele, we're performing monoallelic transfections and assessing through mass spec. So we take a HLA class 1 deficient cell line. We transfect a single HLA allele into that cell. Then we purify the HLA B2M plus peptide complex. We elute off the bound peptides, perform mass spec on those eluted peptides, and finally we're able to identify the sequences of bound peptides, knowing that those peptides came from that transfected HLA sequence. Taking this to the next level, what we're able to do is then develop our own internal MHC binding prediction algorithms. And in this case, what we developed is an artificial neural network, which takes in that mass spec derived peptide data set and then predicts presentation. And we greatly improve both the sensitivity and specificity of predictions over previously developed approaches. The next approach I want to talk about is optimizing coverage of HLA alleles and why this is critically important. So it's been hypothesized that zygosity of HLA class 1 alleles could lead to a more diverse set of antigens that are presented, and really this could potentially correlate with improved survivability for immunotherapy. This makes sense, as the more diverse set of HLA alleles you have, the larger the repertoire of peptides that are going to be able to be presented for the immune system to recognize and target when a patient's on immunotherapy. As Kadar had spoken on, we not only augment and improve the coverage across genes which could potentially turn into neoantigens, but we also improve coverage across HLAs themselves. So here with the same blue-green plot, we see that we dramatically increase coverage across HLA A and B. This is really important because you want to have strong coverage to be able to accurately HLA type, as well as to identify somatic mutations within the HLAs themselves, which can be a tumor escape mechanism. Now, correct HLA typing can be critically important because if you choose the wrong HLA type and you perform your neoantigen predictions, any neoantigen which you predict to be bound to that HLA could potentially be wrong. So if you have 6 different class 1 alleles and you're wrong on 2 of them, potentially, of course this is an oversimplification, but potentially 1/3 of your predictions could be wrong. Now, we performed an extensive validation on HLA alleles across MHC class 1 and class 2. We've done this all the way out to 4 digits. We validated on both histocompatibility working group samples, which are commercially available samples, which have been typed and analyzed extensively in the community, and also on a blinded set of 62 orthogonally and clinically typed samples. We observe a very high concordance. For class 1 and class 2 of this blinded, clinically validated samples set, we observed a concordance of 98 and 94% respectively, with an overall concordance of 96%. And we see that not only do we go after the standard HLA class 1s, but also a diverse set of HLA class 2s. Here's an example of our neoantigen ID sample output. Here we provide summary metrics such as tumor mutational burden and neoantigen burden. And further, we also assess how many of those neoantigens have evidence on both the DNA and the RNA level. Further, peptides are potentially better targets if they're actually predicted to bind to more than one HLA allele. So here we provide for you the number of HLAs that a peptide is predicted to bind to as a summary metric for 1, 2, or 3 HLA alleles, as well as the number of times that was expressed, observed, and affinity range and median affinity values. And we do this for both HLA class 1 and class 2. Here's a list of our top 10 neoantigen peptides based on binding affinity. Here we provide the peptide sequence, the HLA it's predicted to bind to, whether that gene is expressed, the DNA and RNA allelic frequency for that event, as well as NetMHC and NetMHC pan binding affinity. As I'd mentioned, we're developing our own tool internally, which will be available in the future. We also list the binding core, which is used by NetMHC and NetMHC pan for its prediction, as well as other important secondary features such as similarity to self, known antigens, and immunogenicity. And while here I'm just listing the top 10 neoantigens, we also detect and report additional neoantigens, the full set. I'm also going to talk a little bit about leveraging tumor immunogenomics to inform rational vaccine design and patient selection. There are a diverse set of biomarkers for patient response to IO therapy that have been published or discussed at conferences. Here's just a few examples, such as B2M or genes involved in the JAK-STAT pathway. There's also far more complex signatures, such as the IPRES signature, where you're taking and looking at genes expression patterns across multiple gene sets to try to predict response to checkpoint blockade therapy. We report on SNVs, indels, and fusions. And we feel that those indels and fusions really help define the biomarker and neoantigen repertoire. And as Kadar had spoken on earlier, we really have a comprehensive set of readouts for our neoantigen product and immunogenomics reporting, including the list of neoantigens, the tumor mutational burden, MHC binding prediction, and of course the HLA typing. We also list both the expression values and somatic mutations as part of our antigen processing machinery, immune modulators, adaptive and innate immune response, repair and replication, tumor-associated antigens, cytotoxicity, and cytokines and chemokines readouts. This really allows the researcher to very quickly assess and review potential tumor escape mechanisms, and to really look at the total view of how checkpoints could be affected or how well they may work in the patients. I'm sure many of you have seen this picture before. Here we're looking at a publication from 2012, which was really a hallmark publication, looking for objective responders and noting that people that were objective responders to checkpoint blockade therapy were positive for PD-L1 status. Now, we not only report on PD-L1, PD-1, PD-L2, but we really have a comprehensive profile of immunomodulators. In our immunogenomics ID readout, we look at important checkpoint genes such as OX40, ICOS, CTLA4, PD-1, TIM3. And again, we feel that indels and fusions are a very rich source of immunogenic neoantigens. Here again, we're looking at the melanoma data set, which we are working out with in collaboration with the Inova Hospital System. Each dot represents a patient. What we're looking at is the mean number of neoepitopes per variant type. So we're looking at SNVs, indels non-frame shift, and frame shift indels. What we see is that you have a much larger number of neoepitopes that are predicted from frame shift events than non-frame shift events. This is why we really feel it's critical to include both the indels and the fusions into our neoantigen prediction pipeline. Our augmentation improves coverage across other important genes such as B2M, which is important as an antigen presentation machinery gene, and also C2TA, which is a master regulator of MHC class II gene transcription. We also augment genes involved in the DNA damage and repair biomarker. And this is critically important because recently pembrolizumab was listed as a pan-cancer therapeutic for all patients which are MSI high. Here's an example of our immunogenomics ID sample output, where we're looking at the gene symbol, common symbol, general category, gene expression levels, variant type, DNA and RNA allele frequency, variant effect, and that impact across, as I was listing that set of immunogenomics categories, we report this across all of those categories. We provide a comprehensive set of deliverables for our ACE Immuno ID product, as well as our neoantigen ID and immunogenomics ID, including both the raw and processed data files, variant calls, both germline variant calls, as well as somatic variant calls and reporting. We identify events in both indels, SNVs, and fusions, as well as looking at gene expression. For our neoantigen ID product, we look at HLA typing, as well as the neoantigen peptide prediction. And for our immunogenomics ID report, we look at a comprehensive set of genes, the variants in those genes, and the expression of those genes. Now let's take a look at a quick case study where we're able to apply both our neoantigen ID and immunogenomics ID process to be able to quickly look for tumor escape mechanisms in a patient cohort. Here again, we're looking at the 20 late-stage melanoma patients. Based on RECIST criteria, at roughly the 9-month time point, we're able to separate out patients into progressive disease, partial responders, and complete responders. And here on the y-axis, we're looking at the neoepitope burden. Again, based on neoantigen burden, we're able to separate out, for the most part, the complete responders and progressive disease cohorts. However, we have these two interesting cases of extreme outliers with very high neoepitope burden, but a lack of response to checkpoint blockade therapy. Now, applying our immunogenomics reporting, we're able to quickly identify that this patient here in red has a very high level of expression of IDO1 relative to the other patients, and that this patient in green actually has two likely damaging somatic mutations in HLA. In fact, they're in HLA A0201 and 1501, which are known to be broadly tuned. We see a stop gain early on in 0201 and a splice region variant in the middle to later half of 1501, which could affect folding. Now, if we dig into this a little deeper and we look at the predicted number of neoantigens that are associated with each HLA, we see that roughly 60% of the predicted neoantigens are likely to be presented on one of these two alleles, meaning that if you knock out these alleles, you're likely knocking out a large percentage of your presented neoantigens, likely causing a lack of response to checkpoint blockade therapy. All right, well, thank you very much for joining today. We'd like to move on to the question session.
Well, thank you very much for that insightful presentation. Now, I would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of this webinar. We have received some questions, so I'll go ahead and get started with those. And the first question we have here is, what are the sample requirements for ACE immunoID?
I can take that question. So we can work with as little as anywhere between 50 to 100 nanograms of the input material. However, what we must also consider is not just the quantity, but also the quality of the material. So we have a detailed sample preparation guideline, which we can send you. But in general, we can work with a variety of different samples. We can work with FFPE, we can work with fresh frozen, we can work with blood, cells, or just extracted DNA and RNA also. So when it comes to fresh frozen, we usually tend to ask around 30 milligrams of tissue. For FFPE, roughly 4 curls, each 25 mm square, around 5 to 10 microns thick, with tumor content of at least greater than 20%. And for cell lines or sorted cells, we would request 10⁶ cells. And we also can take FFPE blocks, as we have tissue sectioning capabilities in-house also.
Great, thank you for the reply, Dr. Xtalks. And the next question we have here is, what is meant by the similarity to self-score in the neoantigen analytics mentioned during the presentation?
I go ahead and take that one. So our similarity to self module produces a metric for each peptide that indicates how similar the peptide is to any portion of any protein that the subject's normal tissue produces. In other words, the more negative, the less the peptide looks similar to the body. Now we find that if the value is 0, that means the peptide matches a self protein. So it's a perfect match. This is an internal tool that we developed here at Personalis. And the metric is based on running a BLAST-P search using the candidate neoantigen as the query sequence and that patient's own normal as the database to search against.
Great, thank you for that answer, Dr. Boyle. And the next question we have here is this assay appropriate for archival FFPE tissue?
I can answer that question. So usually we accept FFPE samples because that is the most common type of sample which we currently get from our patients. And yes, we have worked with archival samples as old as 10 years, and we can analyze both the DNA and RNA from those samples. So what we have actually experienced is that it's not actually the age that is very critical, it's whether the sample was fixed correctly or not, whether it was stored, that is the most critical part. So if it's fixed properly and if it's stored properly, we will be able to work with even archival samples.
Great, thank you for that response. And the next question is, if I have exome and transcriptome data I've ran using other platforms, do you currently accept this data for your pipeline?
So I can again answer that. So the neoantigen ID analytic engine which we have is optimized to be used with our own personalized ACE immuno ID. Because as I said, since we use the augmented technology, our footprint is slightly larger. It is around 69.4 MB. And we also sequence the match normals along with it. So what we feel is that our algorithms and bioinformatic pipeline is much better trained using our ACE technology. So currently, we would not be able to accept just data from other transcriptome analysis.
Perfect. And the next question we have here is, how does Personalis transfer the data to customer sites?
Usually we offer multiple different options for transferring data. We can deliver data through hard drives, or we can also transfer it through customer cloud storage data buckets. So Google or AWS.
Great. And the next question we have here is, what is the immunogenicity score you report on?
Yeah, I can go ahead and answer that one. So immunogenicity is a measure of how likely the given MHC peptide complex is to be recognized by a T cell. And so increasing positive scores on our metric, they indicate higher immunogenicity. And really critical factors that regard the immunogenicity prediction are the position of presented peptides. So here we're looking at really positions 4 through 6 for class 1. The type of amino acid, so is it aromatic, non-aromatic, acid or base, is it charged or non-charged? And these factors are really part of our internally developed tool for immunogenicity score. It's important to note that here we're not actually assessing the TCR repertoire for this prediction. We're looking at that cell surface presented set of amino acids and trying to say, statistically, are these more likely to be immunogenic or not? And we have enough data that you're able to actually make that prediction and improve your neoantigen ranking.
Great, thank you for that response. And the next question we have here is, do you see neoantigen-based therapies overtaking traditional therapeutic methods as the primary therapy, or rather a treatment that is in conjunction with traditional therapies?
Could you repeat that again, please?
Yep. So do you see neoantigen-based therapies overtaking traditional therapeutic methods as the primary therapy, or rather a treatment that is in conjunction with traditional therapies?
I really see it being a combination. I see that likely immunotherapies will certainly be gaining ground and becoming more popular and more common and more effective as we go forward. But I really see the work that's been presented at ASCO recently and AACR showing that really by working in combination, looking at potentially radiation therapy or chemotherapy and then adding checkpoint blockade on top of that, and then on top of that probably layering in neoantigens and vaccine developments. I think that's where we're really going to be targeting in the future, and that's where the ball is going to end up.
Well, thank you very much for those answers. We have reached the end of the Q&A portion of this webinar. If your questions weren't answered, Personalis will be following up with you, or if you do have any further questions, please direct them to the contact information showing on your screen. And here we have an email address, which is info@personalis.com, or feel free to follow Personalis at Personalis Inc. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is greatly appreciated as it will help to improve our future webinars. Now please join us in thanking our speakers, Dr. Sean Michael Boyle and Dr. Kadar Hasluk, and you can do so by clicking the link in the chat box. We hope you have found this conference informative. Have a great day, everyone.